Okay, so initially David Kleinfeld had asked me to speak last week, and since the subject was more coding, I was thinking of telling you about some recent work, some recent thinking about population coding through a projection, random projections between spaces of different dimensions. But then when we moved the talk to this week, which is more on circuits, I figured I would talk about the retina, and I figured that would be a nice complement to the talk of Tim this morning. And in fact, I think it's not a bad choice because it will be both complementary and orthogonal. Tim was focusing on excitation and spatial aspects, and I will focus on inhibition and temporal aspects exclusively. Now, there's another reason I decided to talk about this, which is much more experimentally centered than questions on coding, is that towards the end of last week, I kind of imagined for myself the following scenario, that there is some civilization on another planet where they have a really good theory of their alien brain. And there's a PI who sends his graduate student to this meeting to say, okay, go and learn about the human brain. So he comes here and then he goes back, and the PI says, you know, what did the experimentalists talk about? And he says, well, you know, we had some very nice, interesting things about C. elegans mating and some surprise responses in the auditory cortex and some large-scale experiments and intricate excitation-inhibition dynamics and so on. And then he says, okay, so what was the theory of the brain? And then, you know, I could imagine the embarrassment of the graduate student trying to explain how the theory of the brain was done and giving various examples. And so for the sake of that hypothetical graduate student, alien graduate student, I figured I would discuss something which is really very experimental, very connected to data. So it's a very simple story, and you can just relax and enjoy it as you would enjoy a movie, if that's the kind of things you enjoy. So
did the PI ask him if it found civilization?
No, it didn't go that far, actually. So this is, whoops, sorry. So this is in close collaboration with the lab of Boton, Roska, and we also used the arrays, microelectrode arrays that are produced by the lab of Andreas Hierlemann, which at the time had about 20,000 electrodes. Now maybe will have 40,000. Antonia is an exceedingly industrious graduate student. She just graduated and is at Stanford now, and she did most of the measurements. And Felix Franke is a very gifted postdoc who did a lot of the theoretical work. And the main story I'll discuss, if you're curious about it, came out a couple weeks ago or so. If I have time at the end, I'll also mention some unpublished work that was done together with Damon, who's at Yale, Damon Clark. So in the introductory talk that Tanya gave, and then in the talk of Eli, both of them pointed out the relevance of looking at different cell types. And Brent also yesterday kind of showed that by considering different cell types in model, one can kind of enrich the understanding we have of the dynamics. And so the question I want to ask is the following: is how should we go about talking about the function of a cell type, in this case of an interneuron, and I'll tell you what I mean by this. So imagine that you have some brain area and you're looking at a given cell type in this area, and you want to ask a question: what is the function of this cell type? So one way you could answer this is to say, okay, I'm going to perturb this cell type, all cells of the same type, and ask how will the behavior change in a given task or a given set of tasks. Now this is, of course, very ambitious, and you could reduce your ambition and say, well, I will perturb a given cell type in a brain area and ask what's the effect on all the cells of the brain. That's still quite ambitious, and so we will look into a more modest version of this, which is to say we look just at a brain area, we consider a given cell type inside this brain area, and we ask, if I perturb this cell type, what changes in the input-output relation of this brain area? And what I mean by this is you can always cut out an area from the brain. There will be projections outside of this brain area that one can think of as the output channels of this brain area, that the brain area sends to another brain area, and there will be some inputs to that brain area. And inside that brain area, there will be this one interneuron type in which I'm interested, and I call it interneuron because it just doesn't project outside the area. And it will influence itself, maybe it'll influence other neurons, other interneurons. It will also influence projection neurons, but we're not so much interested maybe in this detail, but how it changes the output of the brain area. So that's at least one procedural way to think about what is the role of an interneuron. Yeah?
Why are some interneurons not connected?
Because it's a drawing that I did an hour ago.
If you already understand input-output relation of this area with all the interneuron types, why do you want to understand something like about a particular interneuron type?
Right. So
Is it really a meaningful question? What does it mean a role of one type? You have the whole system there where everything interacts with everything. So
right.
you want to understand the whole thing, not the individual type.
So you're right. So if the only interest in the universe was this input-output transformation, what you would do, you could even take a large number of inputs and just catalog them. And you wouldn't even have to understand anything. You could just have a big catalog. Now, I think that while in the abstract this may be correct, I think concretely you learn quite a bit about the input-output relations that you generally don't know in detail if you also understand the mechanisms that operate between the input and the output. That's one thing. The other thing is you may identify mechanisms that are useful to understand other relations in other parts of the brain. And there's a third reason, which is that for a number of, in this case it's very relevant because my collaborator is also involved in medical work, and most eye diseases are actually a deficiency of a single cell type, 99% of the eye disease. And so one thing that you want to do is to say, if I modify one cell type in the retina, how will it change the different criteria you may have on vision? Okay? So there are a number of reasons why you may want to do that. Okay, so I've already divulged that, so maybe for Winfried, what I'm going to ask is I'm going to look into what's the role of an interneuron, and I'll define this by looking at the input-output relations of a brain area. And what I'll choose as a brain area is the retina that Tim gave a nice introduction to this morning. And part of the reason, because as Tim mentioned, that in the mouse retina there are 40 or more types of ganglion cells, and these have different properties, or basically the circuit afferent to them has different properties, and so we'll kind of select different features of the visual input. Now, of course, one possible choice of an interneuron that I have are the bipolar cells that were extensively discussed by Tim. And here's where I say it's kind of orthogonal. What we're focused on will be an inhibitory cell type, and will not focus on the more kind of diverse amacrine cells, but we'll focus on the poor horizontal cell that has got comparatively little attention and is comparatively not understood, I would say. So this horizontal cell in the mouse is only one type. In primates you have two types. The horizontal cell will get excitatory glutamatergic inputs from the cones and feed back to the cones, inhibit the cones through a very complicated, very kind of baroque synapse that is not yet well understood. And there are a number of functions that people attach to the horizontal cell. So one function is that it contributes to the negative surround of bipolar cells. Another function is color opponency, because it will get input from cones of different colors. Only color opponency apparently is not done through horizontal cells in rodents. In primate it's probably true, but not in rodents. Now, it's also believed to be involved in light adaptation, And just to give you an idea of how baroque it is, the reason why people believe it's involved in surround and also light adaptation is they have very strong gap junctions among each other, horizontal cells. So you can kind of think of them as a sheet of electrically connected cell, a big mega cell. And so when you wake up in the morning, there will be some amacrine cells that will release some acetylcholine. This acetylcholine will change the conductance of these connexin proteins, these gap junctions, and so these horizontal cells will become less coupled. So this is completely irrelevant to my talk. It's just to give you an idea that it's quite a complicated system. Now, what we'll focus on is the effect of the horizontal cell on temporal processing. Okay, and we want to understand this, kind of how it will change this output. So basically, yes?
You say it's irrelevant, but I'm just curious, how does acetylcholine make it to the eye?
No, it's released by amacrine cells. There are some amacrine cells that release acetylcholine. It diffuses to the outer retina, and there it.
By light?
Yeah. Actually, so it's by light now. To what I think it's by light now. To what extent it's what the time scale is, I don't know, because it's kind of on a circadian rhythm. So it may be. So I don't know actually what is the mechanisms that. So it could be by feedback from ganglion cells, melanopsin ganglion cells, for example. But I don't know. I would have to look it up. Okay, so the task that we want to, that we give ourselves is to say, okay, I have this little feedback circuit at the outer retina where the cone excites the horizontal cell, which then inhibits the cone, and I want to understand how this little effect of removing the horizontal cell will trickle down the retina. Okay?
It's only the cone.
Well, okay, so that's a very good point, and one of the things that I will talk about at the very end, if I have time, is how horizontal cell contribute to coupling cones and rods. It's an excellent question. Now, everything I'll do here will be in the photopic regime, so it will be in normal daylight, and so there will be no contribution from rods. Okay. So,
they're only talking to receptors. They don't talk to other.
Okay, so that's also a good question. So in salamander, for example, there's evidence that there is direct, and I'm sure Tim may know more than me on this, but in salamander there's some evidence that there's direct information flow that in signals from the horizontal cell to the bipolar cells. In mouse, it's believed that it's not the case, and the only thing they do is feedback to photoreceptors. Okay?
I just want to understand.
Yes.
You said that feedback to photoreceptors, but
yes.
I remember. Okay, so you do kind of a cartoon, but in a way, the horizontal cells are encompassing the synapse between the photoreceptors and the bipolar cells. So they send
Okay.
the reversal potential
No.
at the junction for the bipolar cells.
Okay. Okay, so this is an extremely involved question. Let me just answer one or two sentences, and then we can discuss it, but because it's really not subject matter. You have the cone terminal makes kind of an invagination in which you have processes from the horizontal cell and the bipolar cell. Still in that region, in that synaptic region, so it's a very specific synapse, very, very unusual synapse, and the messaging between the horizontal cell and the cone is not yet understood completely. There are probably three different mechanisms that are active. There's an electrostatic mechanism, a phaptic mechanism. There's a proton-mediated mechanism, and horizontal cell also releases GABA, and GABA triggers a pathway inside horizontal cells, which in turn will regulate these other two mechanisms. That's between just horizontal cell and cone. Now, how is the horizontal cell to bipolar cell transmission in salamander? I don't know. I suppose it's GABA mediated, but I'm not sure. All I'm going to talk about is in mouse. Okay. So this is what Antonia did. So she compared a retina where the horizontal cells are intact, and I have it in blue, these circles in blue, because all the curves that I will show with intact horizontal cells will be in blue. And she compared this with a retina in which horizontal cells are effectively removed. Now again, I don't want to explain in detail how it's done, but I'll tell you briefly. The way you do it is you target to horizontal cells a ligand-gated chloride channel called PSAM, which will be expressed in all the horizontal cells, and then you just inject the ligand, which is called PSEM, and it will basically silence the horizontal cell if you want. So the effect is very similar from blocking the glutamatergic input from cone to horizontal cell. But for the purpose of this talk, you can just think that the horizontal cell doesn't exist, has been removed. Okay? And so what we'll do is to compare these two situations. And as I mentioned, I'm only interested in time, not in space. And so what we'll do is we'll present these two retinas with this stimulus, which is simply whole field stimulus with light and dark contrast of successively greater magnitude. Okay? So this is, oh yes, and I should say, and then as we do this, we record from the ganglion cells in both cases, for both retinas. Okay? So this is, for example, what you get in an on-transient ganglion cell. So it responds to this light increment. And what you see is that when you silence the horizontal cell, the amplitude of the response gets suppressed. Okay? So this is kind of surprising because you're decreasing inhibition, but you also decrease the amplitude of the response. So that's kind of in course the course phenomenon is similar to what the model that Misha did in his childhood, and but it's the mechanism is of course very different. This is the yeah.
77% means 77%.
So this is the detail. This is the number of isomerization per.
Nobody could read that. So go to the next chart.
So this is the contrast. Yeah, in percent is the contrast. It's the Michelson contrast. So the percent is the difference between this light and this dark divided by the sum of the two. Okay. Yes?
So when you are suppressing the small red thing that we see.
That's right. So when you remove the horizontal cell, it's this little curve here.
Okay, I couldn't see that before.
Oh, sorry. Well, you'll see it so many times that you will you will see it. Okay, so basically when you remove inhibition, your amplitude goes down. So that's in a transient on cell. But now, in a sustained on cell, the result is completely different. So in a sustained cell, if you remove inhibition, everything goes up. So the response of the ganglion cell becomes much larger everywhere. Okay, that's in on cells. And in on cells, you also have something that is kind of in between. So in this cell, you can have an increase of the sustained part and a decrease of the transient part. So it's some kind of hybrid between this one and that one.
No, wait, but that one, the hybrid part is bigger.
Sorry?
That's not what you said.
No, so you...
The little red thingy is bigger than the blue.
No, it's not. So here you see there's some blue. Now, I have to say, this is the average. So these curves are averages over many cells. This is an average over 675 cells. This is average over 180 cells over 170. I will show you single cell traces so that you're convinced that this actually occurs. It's not just an average over cells. What I mean by both can happen is that among these 180 cells, you have cells where basically the first transient peak doesn't change and only the sustained part changes. And you also have cells that show kind of this in the transient peak and an increase in the sustained part. Excuse me?
Is there different types of bipolar cells?
So these are ganglion cells. These are ganglion cells. So they're really the output cells of the retina. Yes?
Clarification question. What was again the difference between the three plots?
Okay. So, okay. These are different effects that we cataloged as a result of this procedure. Right. So you take you take a retina, you measure the response to this light stimulus of the ganglion cells. Then you inject the ligand, So that will silence the horizontal cells. You put the same stimulus, okay? And in fact, you do a third thing, which I didn't want to emphasize, but you will wash out the ligand, and you redo the same measurement, and this is this black line, which is nearly equal to the blue line, to check that the retina has returned to its kind of native state, if you want. And then you get many, many different types of responses, and among these, there are some effects that recur a lot, and these are 3 of those. Alessandro, you had something?
No, that I see yellow, so I'm afraid that the manipulation has gone into my cones. I see the tip of the intermediate row to be yellow rather than blue.
Okay, again, these are averages. I will show you maybe one, but I'm not sure the tip of this is the most important message of this slide. So, yes. So, okay, so then in an off cell, which responds to kind of decrements of light, you have the opposite phenomenon. So here by removing horizontal cells, now the response becomes bigger. In the sustained off cell, again, you have the mirror phenomenon as this one. The response goes down if you remove the horizontal cell. And then you have kind of a peculiar phenomenon also in off cells, that in many off cells you have this on rebound. So you don't have a response at the light on, but you have a response with a delay of about half a second. And this rebound on response is suppressed by removing horizontal cells. Okay? So the main point of this slide is to say, okay, you have many different effects that can arise from just removing one interneuron. These occur in transient cells or in sustained cells. Some effects occur in both. They occur in on cells and off cells. And for example, in this cell, in the case of sustained cell, this effect can have 2 different polarities in the same cell. Okay? So it will suppress the transient part, enhance the sustained part. Okay? And so so before I go on, these are single cell responses. Okay, so here, for example, Alessandro, here you can see this one is suppressed in the transient part, so the yellow curve is lower than the blue and is enhanced in the sustained part. Okay, so this is a single cell example. These are not averages.
So you suppress all the horizontal.
Yes. So okay, you can. So you can do a lot of control. It's not 100%, but it's, I don't know, 95 or 96. I mean, and it's kind of uniform on the retina. I mean, there are many, many controls that were done to make sure.
The effects are not very good.
Well, I mean, fine. I mean, there are some effects that are kind of larger than others. So here, for example, you really suppress completely the rebound. Now, I don't know what's your criterion for big or small.
In the cortex, if you suppress the neurons, you have epileptic seizures. So nothing like this.
No, in the cortex, if you suppress inhibition, you have epileptic seizure. This is one inhibitory cell among maybe 50 inhibitory cells in the retina. Yeah, you have amacrine cells. Yeah.
But you block all horizontal cells, right?
Well, up to experimental accuracy. Yes. Okay.
Is there any particular disease that corresponds to this?
I am sure that I don't know a good answer to your question. I am sure that there are some diseases. I'm ready to bet, let's say, that there are some diseases that are correlated with deficiency of horizontal cells, but I don't have a good answer as to which.
Okay, I was just curious what people see under that condition.
Yeah, that's a very good question. So, okay, I can give you another example afterwards, but not involving horizontal cells. Okay, so basically our task is to say we remove this one element in this little loop, and we want to understand how this zoology happens. So what I'll do is, the main part of the talk is to develop a model of the retinal circuit and to try to derive what are the function of the horizontal cell given that model. To do this, I'll have to do a detour and kind of develop a model for the photoreceptor, because that will be essential just to build the model for the whole thing. And at the end, if I have time, I will kind of expand into some, beyond this question of the effect of horizontal cells. But I don't know if I have time. Okay, so we want a simple photoreceptor model. And so the way we'll go about it is we'll say we want the model to capture a number of things. So we want the model to capture responses to flash. So this is a responses to flash of increasing magnitude in the dark. This is the hyperpolarization response of the photoreceptor. We also want the model to satisfy some properties of adaptation. So here you have you're not changing the magnitude of the flash. You keep a constant flash, but you change the background light from dim to bright. And what you see is that the shape of the response changes from monophasic to biphasic, becomes sharper, the peak moves to the left, there's a second bump, and so on. And here, for the purposes of illustration, all these peaks were normalized to 1, to the same size. In reality, their amplitude goes down like 1 over the background amplitude, the Weber law that you all know. And so we want our model to satisfy these kinds of constraints. And so the model I will write in terms of the response of the photoreceptor, which is a deviation of the potential from the dark potential, okay? And so the simplest model I can write is just a linear model where I'll say that the response of the photoreceptor follows a relaxation equation, so it's triggered by some light input. And here y is not the light input itself, but some smoothed light input. So the light input convolved with a filter which has a time scale of 100 milliseconds or so, 120 milliseconds. Now, of course, this would not give me any of this nonlinear behavior, this compression, this plateau, and so on. And so I'll just add a very simple term, so a term which kind of controls the relaxation here, which will also be a convolution between some filter and the light input. Okay? And the second filter, kz, will basically be the same. It will be somewhat slower, and that's it. But it's on the same order of magnitude. So one way you can think about this model is that the light input comes and is divided into 2 channels. And this second channel does not affect directly the response, but kind of tunes the time scale on which the output of this channel is low passed. Okay, that's one way to think about it. Another way to think about it is, imagine you divide both sides of the equation by this term. You see that this term will modify both the time scale and the gain of your response. Okay? So this model is quite good at reproducing the data. So this, for example, is the set of responses to flashes of the same intensity. And what was kind of cute here is that we pushed it to a very large flash. And what we found is this double hump response. And then later on, we found in the literature that if we really give a big flash to the photoreceptor, indeed, you find this kind of double hump responses. It also captures this change in shape of adaptation. And of course, it captures the Weber law very well. So now,
where is the background? How does the background enter?
Because here you have light intensity.
So it's all
Yeah.
including...
Well, the photoreceptor doesn't know what is background. Photoreceptor just knows how many photons are coming in.
Kind of constant thing plus the plus.
Well, you take anything you want.
There is no space here.
There's no space. There's no space in this talk.
And how do you think of Ky and Kz in terms of biochemistry of the photoreceptor?
Okay. No, okay. So you can go back, look at the phototransduction cascade, and you can kind of reinterpret these terms in terms of that. And this will be related to phosphodiesterase concentration, if you insist on doing it. But I don't want to do it now. So I just think of it just...
Different ways to filter, looking at that cascade,
Yeah.
you'll see 2 different...
No, actually, no, that's a good point. So here, actually, so first of all, I should say, this is just a phenomenological model. So I don't even really want to interpret it in terms of biophysics. If you insist that I interpret in terms of biophysics, part of this KZ will come from phosphodiesterase dynamics. Part of it, I think, is just mimicking some kind of feedback that is actually more complicated. So you have some various feedback loops that involve calcium, for example, in phototransduction.
But somehow you wrote down this equation.
Yes.
What was the motivation for writing the second part?
It was,
I mean,
I wrote it.
you wrote it,
Yeah.
it matched the data.
Exactly. This is exactly how I did it. Okay, so okay, so now before going to the talking about the whole circuit, I just want to mention a couple properties of this model because they will kind of come back later on. So one thing is that you see this is a nonlinear model. It's nonlinear in the input, not in the output. But because it's nonlinear, it will have asymmetric responses to on and off stimulus. So here, for example, we give us a brief step of either light or dark, and for light you have a hyperpolarization, and you have a depolarization for a dark step, but it's much bigger. So you don't have an on-off symmetry. Yes?
What's the relation between alpha and beta to get that law?
So, okay, so alpha is basically just a choice of units, okay, because they will convert your light intensity into a potential. And beta is a numerical factor that tells you how compressive your responses are, okay?
So you optimize for
yeah, so we fit actually very few parameters. We fit, basically each of these filters has 2 parameters, I think, and maybe 3. But basically you can fit them by hand. Also, essentially it's beta that is the problem, because everything else you can fit to linear, pretty much to linear responses, and you can fit for, let's say, this, and then look at this. So it's kind of, yeah. Okay, so here it's not, you know, profound, but it's, I will mention this again, so I mention it here, that one automatic outcome of this nonlinear model is that there is some on-off asymmetry in the way these cells respond. There are other kinds of asymmetries, but this is in the model. I don't know if it has been explicitly measured like this. I think probably one can find, actually there's one old measurement, I think, that was done with steps of light that shows it. I can dig it out if you want.
This is a model for an isolated receptor.
That's a very good point, exactly.
It's not just measuring, you would have just to suppress all the retina.
Right, so one way of doing this is to cut it out. So you can do it in slice, and in slice you kind of cut out a lot of the feedback that gets to it.
I think that you fit this model to a receptor in the retina.
That's right. So but again,
Plus all the rest of the retina.
no, so as I just mentioned, in slice you will remove a lot of the rest of the retina.
So these traces were in slice?
Yes, yes, absolutely.
So this was pure receptors?
Pure, well, up to, let's say, quasi-pure receptors, okay? So with much less feedback from horizontal cells, for example. These were these are old measurements, sharp electrode measurements in slice. I will actually show you the difference between slice and whole mount recordings if I manage to, but I don't think so. Okay. So now this asymmetry has also cute consequences, so I'll just spend a couple of slides on them, and then I'll go back to the circuit, is that it influences the responses to fluctuating inputs. So imagine here I show you some fluctuating, Gaussian fluctuating input, and I suddenly keep the mean the same, but change the variance and make the variance much smaller. And the nice thing about this model is that you can trivially solve it for any input. And so here, for example, this is the solution for a flicker like this. So you have the solution, the response that you have to the mean, and that's get corrected by a term that depends on this flicker, where the sigma is the amplitude of this flicker. And then you have this complicated term which involves the background light level. It involves these factors Q's. that are kind of overlaps between the filters, the KY and KZ filters, and chi, which is the correlation function, the temporal correlation function in the noise. Okay? And so depending on delicate adjustments between background light level, the Ks and the Chis, this can be positive or negative. And so flicker can have a boosting or a suppressing effect on the response. So for a given set of parameters, this is the kind of mean response you get. So when you increase the variance, you have a bump of activity. When you decrease the variance, you have an underswing of activity. And the cell is more depolarized when you have more variance, and is more hyperpolarized when it's more deterministic. Yeah. Yeah. And so the same is true for naturalistic stimuli. Just naturalistic stimuli, the fluctuations are much bigger, so the effect is much stronger. And the last thing maybe I want to mention, just to give an intuition about the behavior of this photoreceptor, is if I take naturalistic stimuli, so these were measured by Van Hatteren by putting a camera and walking in the woods, and so you have much bigger fluctuations than the simple kind of Gaussian fluctuation we look at here. And now let's say I want to probe the photoreceptor, so I add a flash at different moments, and I ask what's the impulse response of the photoreceptor. This is what I get. So depending on where I add the flash, I will change the magnitude of my response by 1 or 2 orders of magnitude. And on average, my photoreceptor here is more sensitive than what it would be if I just had a constant mean matched input. Okay? So all this has to do with this non-linearity in the behavior of the photoreceptor. So let me go back to, you have a question?
Yeah, just to go back to the same picture. So, I mean, as you go from the weak responses to the strong responses,
Mm-hmm.
in the white peak, is that going from strong stimulus to weak stimulus?
Yes.
Or is there some kernel you have to average over?
No, it's going from change in the background light level. So you're walking in the woods, there's some change in the background light level, and now I add on top of that small flashes to probe your photoreceptor, like linear response theory, if you want.
But is it, okay, but is there something simple, like the response is proportional to the background light, so you get this?
Yeah, it's inversely proportional to the background light. It's Weber's law. It's Weber's law. Yeah. So basically, what I'm saying here is that because you have these two filters, the photoreceptors is adapting dynamically. So it's as if you're adapting every 200-300 milliseconds. You're readapting your kind of the state at which you operate. Yes.
I'm confused. I mean, basically, you're saying in bright light, you're shutting down.
Right. So what I'm saying is, imagine you take a constant flash of light, of a constant magnitude. I send 10 photons in your eye, but I do it in a dark room, in a slightly more lit room, in a slightly more lit room, in a slightly more lit room, and so on. Every time the response to these 10 photons will be some curve like this, some pulse. The magnitude of this pulse will be proportional to the inverse of the light level in the room.
But I thought Steve Backus showed both ways. I mean, one was adapting, like you're saying, but the other was adapting more about hedge.
In ganglion cells. This is in photoreceptors. Yeah. So in ganglion cells, yeah.
And one more time, dashed and solid red line.
Solid red line is the mean of the average of all of these. Dashed is the response you would have if you had a stimulus here which were constant but with the same mean as this one. Okay, so this was a detour to kind of construct a model for this unit that we need here to then, that will be the input to the rest of the retina. And what I will want is just by changing this one feedback loop here, I want to retrieve all this zoology of effects. And so for this, of course, I need some amount of diversity in my model of the retina. And as we heard, there are on cells that respond to light increment, off bipolar cells that respond to light decrement, and different bipolar cells come with different time scales. So what we did here is we considered simple kind of ad hoc bipolar cells that were inspired, if you want, by real bipolar cells. So one fast one, which basically takes a derivative of its input, One that has a kind of slower derivative, or if you want, is in between a derivative and an integrator, and this is just integrating with an exponential leak. Okay? So you could think of this as a very fast transient bipolar cell, kind of slower transient, and a sustained cell. And then it's rectified, as in the models of Tim, and then we sum them. We sum this input into the ganglion cell, and this, after rectification, will give us a spiking rate. Okay? Now there's one more twist to this, is that it's known that ganglion cells are sensitive both to the input they get and to the change in the input that they get. So we'll add just one more possibility, parameters where this sum can be can just be the linear sum of these inputs, or it can be its derivative. Okay, and this is controlled by a parameter here. I'm not sure you can see alpha, which can be 1 if it's just a derivative, or 0 if it's just a linear sum.
Did you try to
Yes.
to do this also in kind of conductance-based model?
No.
To see differences?
This is complicated enough. No, so you could do it, of course, if you had a good reason, I would do it. I'm not sure I would see a good reason. Maybe if you have a good reason, you can tell me, and I could see why it would be worth. But for now, let me try this simple model, and then you can tell me if you think there are good reasons to go beyond.
So one reason is that you will automatically have sensitivity to function and its derivative.
Right, sure. But so that's right. So one thing is, of course, I could write a conductance equation here and so on. I wanted to keep it as simple as possible and to say, okay, I know this exists, and let me capture this in this simple way, not worry so much about mechanistically how it's done, but to keep kind of morally what's happening in the cell.
So this is which model is this out of?
It's our model.
It's LN or?
No, it's okay. So this is, so you could, okay, this is L, this is N, this is L, and this is N. So you could think of it as ln. But I would say the main twist here is not, how many non-linearities you have. I think the main question is you're changing one element, and you want to see how a diversity of effects can give rise to here. And so what is happening here is maybe the main focus, and the diversity of these channels that I'll discuss more.
There is no space, it's like 1 pixel.
Right. It's not 1 pixel, it's whole field. So the stimulus is whole field, so we did also spatial stimuli to kind of evaluate the influence on the surround, but it's a less interesting story, so I prefer to focus.
I want to go back to my first question. So before you manipulate something,
Yes.
how would this model be? Why would I care about manipulation?
So you will see, because of course I will ask this model to reproduce not only the manipulated retina, but also the intact retina.
So it's a good model of retina.
Okay, so,
The intact model.
right, so there is no, I don't think, if we asked to people in this room to give us a definition of a good model, I have very high doubts that everyone will give the same definition. So in our case, what are we looking at? What I'm looking at is a model that can give rise to the qualitative behaviors that we see in different types of ganglion cell for this stimulus in both the controlled retina and the kind of manipulated retina. Okay? This will be, for me, a good model.
So this means that, okay, you restrict yourself to full field, so
Yes.
say for any time course of full field stimulus, you get the good.
Yeah.
But you have some proof, like some illustration of this, or you just believe you?
Well, that's the point of the rest of the talk.
Okay.
Yeah. So how does it work? So here, this is what happens to this outer retina. So just this module here. So what you see is when I remove the horizontal cell, a number of things change. The baseline of the response changes, the whole thing shifts down, and then the shape of the response also changes. Okay? And then by filtering this trace through these different filters, I kind of select different features of this trace. Okay? So these, excuse me? All this is model. All this is model.
But where is the horizontal cell in the phenomenological model?
Okay, so here the horizontal cell is, you can do a more complex thing where you have also non-linear feedback. In our case, we just took some linear feedback. Okay?
It wasn't in the original model.
Okay, so that was just this.
Oh.
That was just the cone, and then you add this and you add everything.
But it worked so perfectly. Because it was
In slice, exactly. Okay, so basically by these different temporal filters, you kind of select different features from this trace, okay? And then another key point is...
I missed how you do H model. How you do H cell?
Okay.
Is there a filter? That's the question.
Yes. So there is a filter. So there. Okay. So there are various ways of doing it, but the simplest way, which gives you good outputs, is you take the input to the horizontal cell is the cone output, and the horizontal cell is just an exponential filtering of this cone output. You have two parameters: the gain and the time scale. Now, because of this worry of parameters, and in the spirit of parsimony, and also kind of inspired by data, we restrict. We say each ganglion cell will get at most 2 of these pathways as input. Okay, so we'll not allow more than 2. The issue of fitting in parameters on is important. I will comment on it in a moment. Okay. So, for example, if you have one ganglion cell that has these 2 pathways, a kind of a soft transient and the sustained pathway, this is the kind of output that you, from the on bipolar cells, this is the kind of output that you get. And in fact, this is exactly one of the behaviors that we want to reproduce, which is that you have enhancement of the response throughout the on stimulation. And in fact, by combining these pathways, you can reproduce all the experimental effects. And in order for Misha to compare, this is the experimental trace, this is the model. So of course, the details of the shape, here we just wrote down filters arbitrarily, just kind of sums or difference of exponentials and so on. We didn't worry about having a rise time which was fast and a decay time which was slow and so on. So we did this extremely brutally, but I would say given the brutality of the procedure, the comparison is not so bad.
So you only looked at flashes, you didn't look at more general patterns?
No, this experiment took about 7 years to create these retinas. It's a very, very complicated experiment. So, yes, but by then the graduate student is ready to graduate. So,
keep the mice, right?
Yeah, of course.
So then the next graduate.
Well, they have to. Okay. So let me see if I want to comment now or later. Let me comment on the fitting procedure after. But okay, let me comment on the fits after. So basically, we have this model that kind of this simple model of the retina. Now, how do each one of these arise? So for all 4 of these, you only need the same pathways, basically. So this transient pathway and the sustained pathway for the sustained cells. Okay? And now about fitting, what I should mention is that the cone part, so this part is completely, we didn't even fit that part. We fitted that part on cone data that were separate from these data, and we never refitted that model. And for these fits, the filters we didn't fit. The only thing that we fit was the prefactor, and we chose numbers like 1, 2, or 3. So we fit completely by hand So there was no actual fitting procedure that we followed. Okay, so all this is got by this method. Now, one peculiar example is this one, which I will discuss in a bit. And this one has a fast off input and has a sign flipped intermediary transient on input. And the way we interpret this sign flip is just to say that this had to go through an amacrine cell. So it's kind of an inner retina unit, but it's not pure bipolar input. It has a direct inhibition from amacrine. And this will be important also. And the last thing that the model says is that if you only have fast input to the ganglion cell, then horizontal cell has no effect. And indeed, there are cells that are like this. So I think I kind of tried to argue that there are, in fact, not so many parameters that are fit, and the fitting is done really very brutally by hand by choosing these prefactor to be 1, 2, or 3.
Yeah, but it's not, I mean, you have 6 choose 1, 6 choose 2, 6 choose 3. I mean, we're getting up to possible pathways. I mean, where are we up to, about 60?
No, because...
possibilities that you're...
No, it's...
saying there's very few parameters, and then you're just picking pathways.
No, this is not true, because because there's only one sustained cell here, and here, if the cell is sustained, it will have to get input from this one. Okay. So I mean, it's much more constrained than you would think. If you start thinking about it, if you say yes, I have 6 pathways, but these are off pathways, so you can throw them out. And so if you start thinking about it, it's actually quite constrained. It's surprising. If you actually do it, it's kind of surprising that it works so well. You wouldn't expect it. Now, because I knew that there would be this criticism, because the kind of criticism you just said is exactly what I would have said if I were sitting there and somebody were presenting this. I would have said exactly the same thing. And so my way of answering, instead of kind of arguing for this, is that I will show you that you can make predictions with this model and then you can verify them experimentally.
So this rebound response, where does it come from?
So what the model says, and I will show you experiments, is that, can you wait just 2 slides, because I will explain it on a couple slides. I will devote...
There is no simple answer.
There is, but I want to give it in a couple slides. There is actually, because I want I have a slide on it, so let me get to it. Okay, so let me show you a few predictions. First of all, let's take this model and say, okay, let's believe in it. What does it imply? Make some predictions and then test them experimentally. And we actually did this. So one thing is you can plot the magnitude of these 2 curves, the blue and the yellow curve, as a function of contrast. Okay, and what you see is that in some cases like this one, there's a shift. In some cases like this one, there's a gain change. And I can here draw scatter plots of these 2 curves. And I will see that, for example, in this case, when I remove the horizontal cell, I compress my dynamic range going from here to here. In this case, I expand my dynamic range going from here to there. In this case, I just shift my dynamic range. Yeah.
Essentially, you have 2 conditions.
Mm-hmm.
So when you fit your model, you fit it to one condition and then test on the other, or do you just fit it to both?
Again, I mean, it was much more childish than this. You just, basically, you have a few parameters, you just pick and you try and you see that it works well. There's no fitting. We never did fitting.
So when you see that it works well, you see that it works well for both conditions or for only one?
Well, you can see yourself. This is what I mean, but it works well.
Both conditions?
Yeah. Okay. So...
Because one way to get around the large parameter set is to say that we only fit it for one condition and then see that...
That's right. But we don't fit, so... Okay, so if you... Yeah? What? Sorry?
You do fit. You just fit by eye or something.
Oh, okay, well...
You cannot just swap parameters and make it work. You never do.
I think they're
Well, that's what we did. What do you mean? That's precisely how we did it.
For all parameters in rho, you get whatever.
No, of course not. I told you, we just say the parameters will be 1, 2, 3. We just pick those and we just put that, and this is what we get.
Yeah, but I guess you look, let's say, at the decay time of these responses. So you put
Okay, so what? That's right. So you have to. So
you are saying there is no.
no, I never said you don't think. I never said you don't think. Of course, you have to think about it, and you can see what makes sense. But then you just have to fit. Okay. There's no objective function, none of that. Okay.
But you made it look similar to the data by kind of putting some reasonable.
In other words, you could improve the test.
That's right. We could, but that's not our aim. Okay, that's not our aim. I think that's missing the point to think in this direction. Okay, so this is what you get. Okay, so you get a pretty good agreement for a model that was not fitted.
So now you understand now it works, but do you understand why it works? Why here in the trace above the bottom, the red is above the...
Yes. So actually you understand every single one of them. I thought it would be a bit boring to go through every single one of them. I will show you some example, which will answer Misha's question also. Okay? But if you want, you and we can go through, I understand each one of them. Okay. So let's come back. Let's come to this. I hope will answer another prediction concern this kind of peculiar case of this off cell. And I think this maybe answer part of your question, maybe answer Misha's question. So how does this rebound thing work? So the only way we could think of it working is to say that here in this pathway you see this decay, the off part of the response, the decay part of the response is much steeper with the horizontal cell than without. Okay. Now if this comes in your ganglion cell as inhibition, this now will become a positive slope. And if your ganglion cell is sensitive to changes, it will be much more sensitive to the blue than to the orange. Okay? And so we think that this is what generates this rebound in the ganglion cell. So what does this imply? This implies that there is direct inhibition to the ganglion cell, which direct inhibition does not need to be the cause of a rebound. In fact, typically one would not think of direct inhibition as causing a rebound. And also what you want to see is that the inhibition this slope becomes more shallow when you remove the horizontal cell. Okay? And this is what Antonia did. She patch clamped these cells. You see that the rebound in these patch clamp experiments also is, you see it's suppressed. These two traces are the traces before the chemical manipulation and after washout. This is when PSEM is bound to PSAM, and these are the two measures of inhibition. Okay, and what you see indeed is that this curve becomes much more shallow at the moment of the rebound. Okay, so this. It may not give you shivers, but I can tell you that it gave her shivers because she was very surprised that a model at this level of simplicity could actually suggest an experiment to her that she would do patch clamping and find exactly what the model says. Now, my whole reason at the very beginning to mention this on-off asymmetry in the cone was because of this. So another prediction is the following. Because of the on-off asymmetry that we have here, what you see is the response. These are response in the cones, but where I've subtracted the baseline just to emphasize the difference between the yellow and the blue line. So if you look at this, you see that the difference here between the yellow and the blue, or the orange and the blue, is much bigger than this. Okay? So in the on part of the stimulus, you have a bigger effect on the peak of the removal of horizontal cells. What? Okay, look at here. Here there is a difference. The orange is here, the blue is here. And here there's no difference.
But there is a bit later than here.
Okay, but I'm looking at the peak.
Only looking at the peak.
Yeah.
So all this is because you're getting, I mean, you're basically changing the response of the cone because you have some feedback inhibition with a slow tail response. And
No.
It's enough to isolate a peak.
No.
I mean, that's what you've done. It's just one. It's very simple. You're feeding back a low pass filter.
No. So the feedback is linear. What I'm saying is, I'm saying something else. I'm saying the difference between these two curves here and these two curves here are different. The difference is different. And the reason it's different is because the cone is nonlinear. Had the cone been linear, these two curves would be mirror. The on and off curve peaks would be mirror images of each other.
But the cone is nonlinear because of
Because the model of the cone is nonlinear.
in a retina model.
The cone is nonlinear because the cone on itself is nonlinear. Erase everything else. The cone on itself is nonlinear because it has gain control and so on. Yeah, that's that model. Exactly. That's why I emphasized the nonlinearity in that model was all to get here. Okay. So now what does this say? This says that if you have a pathway that will pick this peak, that will select this peak, you should see a much bigger effect of PSEM on the on cell than on the off ganglion cell. And this is indeed what you see here. Here the red is on cells, the blue is off ganglion cells. This is the response in different cells when in the control retina, and this is the response after manipulation.
I'm a bit lost about this kind of the whole approach. So you made a lot of effort to model the cones,
Mm-hmm.
right? And you have an experiment to validate
Mm-hmm.
the model. But with the horizontal cell, somehow you just say, okay, this is just linear filter, and you don't show any evidence that.
So, okay, we have. Okay, you have independent reasons to think that that's not a bad approximation. Now, from other data. Now, the cone, there are immense amount of recordings from cone. These kinds of nonlinearities in cones are extremely well established. There's no way out of including them in the model. Now, what you say is probably true, that if I make my horizontal cell feedback also nonlinear, probably I can get better agreement with the data and so on. But in the spirit of parsimony...
There is no data on horizontal cells.
No, of course there is data, but much less than on measurements of cones. And what I'm saying is that, yes, probably if I go to those kinds of measurements, I would probably want a nonlinear model, and it would make my model more complicated and so on. But again, what I want is to kind of capture a whole set of phenomena and understand how they emerge, rather than have excellent fits for each one of them.
No, I understand the goal, but why is it important to model cone cells well and not important to model horizontal cells?
No, it's important to model everything well. But for the cone, because for the cone model, if you put a linear cone model, then many things will go wrong very fast. And if you, okay, now maybe there is a way to have a strong nonlinearity in the horizontal cell that would kind of mimic also that nonlinearity in the cone. But since you know that the cone has this gain control and so on, yeah, it's natural to put it in, you see. But I agree with you that probably if we, for example, push to higher light amplitude and so on, maybe we will need to include nonlinearities in the horizontal cell. Okay. So anyway, so this is kind of, again, an agreement that the on pathway will be much more sensitive, and at least within the framework of this model, we trace this back to nonlinearity in the cone. So that to me was kind of amusing, because it means that some effect on ganglion cells that distinguish on and off ganglion cells can be traced back to adaptive properties of the cone. okay, which I don't think kind of when you wake up in the morning, it's a thought that comes intuitively.
Horizontal cells, they talk, is it indeed like you have one horizontal cells talking to just one cone, or it's a...
Okay, so this also, again, there's a complicated answer, but I would say the default view that you could have is that horizontal cell is a very large cell because they're connected by gap junctions, okay, to each other. And if you have a whole field stimulus, you could just think, okay, every cone is getting kind of the same feedback from the horizontal cell sheet.
Kind of giving some negative feedback to all of the,
Yeah.
all of the cones.
Yes.
But you only, well, you ignore this.
I ignore because it's spatially uniform, so I don't, yeah. Okay, let me skip over these, and okay, maybe let me just mention this. So again, all of this was kind of based on this, I just said that this depends on this change of shape that we get in the cone. Now, we didn't measure the cone.
Actually, this would predict that if you make your, because basically this, you say this kind of giant horizontal cells is excited by all the cones. Okay, let's say this approximation. So
okay.
This would mean that if you make the size of your stimulus bigger and bigger, there will be a big change because the horizontal cells will get more and more interested.
No, of course. And many studies of the cone are based on these kinds of things. In fact, in this work also, we looked at spots of bigger and bigger size. But I'm just obliterating. Yeah. But I'm kind of putting this aside because I think it's less. You're right. Actually, I think along that mindset, a more interesting question is, what about if you have a big bar moving? Yes, you're electrically coupled, but there's still some time. There's a time scale associated with how your potential will change in that sheet. And is there some interplay between the movement of the bar and how the potential changes in that sheet? So it's kind of, I don't think, for example, this experiment has been done carefully. Anyway, so if you take calcium imaging of cone terminals and you kind of deconvolve to try to find the potential, this is what you get, comparing the native state and the SEM state, and it kind of looks again like the prediction. So that's another kind of, if you want, microscopic validation of the model. So let me So I this is close to the conclusions, at least of this part. So I called this talk like the 6 functions in search of a interneuron, and the Italians maybe understand why. And the reason I was thinking about these 6 effects. Now, these are not really function, they're really effects. And well, there are 2 here, because there's this peak that is changing and this rebound. But well done for the counting. And so now these are effects, they're not really functions. Now, if I want to speculate on what function are these fulfilling, maybe one could say the following, and these are really speculations. First of all, you see that there's some temporal sharpening due to horizontal cells. So with horizontal cells, you get these temporally sharpened responses. And what's kind of unusual, and that's really has to do with the LN-LN nonlinear structure, is that when you have temporal sharpening, generally you have lower gain. So you have a gain bandwidth trade-off because you sharpen temporally by removing the response to some inputs. Whereas here, you sharpen temporally, but you boost the response. So it's kind of unusual, and it's not kind of the same thing as linear temporal sharpening as you would think. The other thing is, in terms of number, most of the cells had their... sorry, I have this wrong. It should be... I have it inverted. It should be this one. Anyway, sorry, this drawing is wrong, so let me say it in words. Most of these cells are... The dynamic range of the cells are boosted by horizontal cells. So for 70% of the cells, if I remove horizontal cells, their dynamic range goes down. Okay? And the third thing, which is curious, is this rebound here. Now, This rebound is like a signal, like a memory signal that you have half a second after a light increment, but in an off cell. Okay? So it means that if now I give an off stimulus here, I would kind of have an enhancement of the off response. So for example, if this would give you a mechanism to get tuning, if you have a moving bar, for example, this cell would be tuned to a specific ratio of the size to the speed of the bar. Okay? So that's a very naive guess of what this kind of rebound response could be used for, but this is just a naive guess. So maybe I stop, or yes?
If you have intact circuit, can you trace differences between on and off ganglion cells to the cone non-linearity? Like sharper non-linearities for off neurons than for on neurons?
I'm not sure I understood. So yeah.
People study differences between responses between on and off cells.
Mm-hmm.
Off cells usually have sharper non-linearity than on cells.
Okay.
And I was wondering whether this can be explained by nonlinear cone dynamics. So you put in a white noise.
No, I understand what you're saying.
White noise, you measure the nonlinearity.
It could be.
And
no, I understand what you're saying. I think qualitatively, yeah.
If you just have a cone and so on going to ganglion cell, on and off cells, one will have a sharper nonlinearity than the other.
I think qualitatively the answer is yes. What I worry about is that you have the ganglion cells will be faster than the cone. Now, the one way to do this is to have kind of derivatives in between. And so what you would need to do is to take a model like this with derivatives, with like transient pathway in the middle, and do your analysis, and while keeping the nonlinearities of the ln nonlinearities the same. And my guess is that the answer would be yes. That's a nice thing to try. Yeah.
Is the horizontal cells basically turn off in the dark? Do you know about light? I mean, if you adapt to a very dark room to a very light room.
I'm not sure what you mean by turning off.
I
So
I thought there was something that the gap junctions. I mean, the gap junctions change with dopamine.
right.
You pointed at the beginning.
Right. So
they change with sustained light dark. Because the problem is from detection to discrimination. When it's very dark, you just want to know if there's a bright object.
right. The problem is that what I said before kind of goes against this.
Well, no.
But I think it, as far as I can tell, it goes in the wrong direction in terms of the connexins. Now,
because I look at the responses,
yeah.
I mean, the responses are in the net area under the curve in some sense, without the yellows, without horizontal cells are greater, so that should increase detectability.
First of all, it depends on which ganglion cell. These are ganglion cells. I'm not sure I can do your argument here, but you see, in light you would want a bigger surround. What you have in mind is the spatial surround. This is all temporal, but in light condition you would want a bigger surround. Now, if I remember, temporal also, but what I remember of the connexin business is that the coupling between horizontal cells decreases in light rather than increasing, which is weird. So maybe I don't remember correctly, but I think that's what's happening now. There's a lot of gain control already in the cone, so I'm not this. Yeah. Okay. So who asked me about rods? Someone asked me about cones. You asked me?
No.
Misha asked me about rods. No, somebody asked me about rods. Okay.
What about rods?
Okay. So let me just show you this and then I finish. I won't say the last thing. So these are, but this relates to your question about slice and not slice. So these are measurements of cones, and you put a step of light with a given contrast, and you increase the background light level. So at night, the cones are not functional. In the day, they're functional. This is for rods. So rods work at night, they don't work in the day. Okay? And you can generalize a little bit our model to fit the rods. Okay? Now, this is in slice. When you measure in whole mount retina, so when the retina is intact, this is what you get for the rod. Okay? So what you see is at night it's kind of similar, but then when the cones kick in, you get this big depolarization. And so an obvious guess is that there's horizontal cell coupling between the two. And so it will flip the sign of this, and you get that. Okay? And you can do this model and then fit each one of them, and then you're left just with one parameter to fit for your horizontal cell, and you get kind of a nice agreement. Okay? And you get even these little blips here. Anyway, so let me skip this part on adaptation. Okay, and let me conclude. Okay, so I think one main conclusion, which is absolutely obvious, of course, is that a cell has a very different influence on its postsynaptic cell than on the rest of the circuit. This is an obvious statement, but it's kind of interesting, it's kind of amusing to see a specific example of it. And also it can give rise to a big zoology of effects. And unlike what was the belief until recently at least, it is possible that horizontal cells have a role also in kind of temporally chiseling the response and not only in spatial surrounds. And then finally, I didn't have time to show you the work on adaptation, but I think one value of these simple models that you don't have to fit are not only that they can reproduce data in a coarse way, but also that they give ideas of how to generalize. And kind of by making them a little bit more complicated, you can kind of explain phenomena that are observed, but you probably wouldn't have thought of these explanations without them. And so I'll end here. Thank you very much. And this is what it looks like, cones and horizontal cell. Okay, thanks. Okay.
So, I want to say something about the difference between the model you're using, and the more biophysical model.
Mm-hmm.
So, one problem with this model is that, in a sense, they are made up. They may reflect the activity of single neurons, maybe, but you need to show that. Otherwise, they are not. You say, okay, this is a system. There is a system that can replicate it, but you're not sure that the system that you use really, the components of it are the component of the real cell.
That's right.
Number two is there are some experiments for which you need to explain you must have some relation to biophysics. For example, if you have a drug like this that blocks this current, that current.
Sure.
And you cannot do it with this, because how do you know what is the...
I don't think it's as clear as that. So because I know I get these questions, so this is, I'll just 1 minute. This is, if you want a biophysical, this is a cone or a rod. You have the pumps, you have the channels. This is the forward cascade. This is the equations for the forward cascade. These are, you have feedback loops involving calcium, you have feedback loops not involving calcium, you have all kinds of biophysical mechanism, diffusion, and so on, to take into account. And you need at least as many parameters that are not measured. Okay? This is only for the cone. So I'm saying, of course, ideally you would want a biophysical model of the whole retina. I think there are two issues. One issue is that there are several issues. First of all, in this there are probably many of these feedback, and you have feedback of, control loops on control loops on control loops that are not useful for describing the answers to a broad range of stimuli. So you may not want to take them into account. That's the first thing. Second thing is you don't know what the parameters are, many of the parameters. So you're back to fitting. And then the third thing is that it's very hard to think about them. It's very hard to kind of develop intuition when you use these models.
There's this way of simplifying model.
Yeah, the sloppy stuff.
Yeah.
I want to. You know about it? You can explain it to me. I haven't looked at it in detail.
Maybe, I just heard about it.
So this full field stimuli are not interesting, right? Because that's not visual. So why bother with the models before they work?
I think no, you're right. So I think well, I think you have to start somewhere first of all, and I think that now I'm maybe not myself so interested, but the postdoc is quite interested to look at making it spatial. But at least now you have some.
No easy way to go from this to spatial. You will have to really deal with all these issues.
No, of course there. No. Of course there is an easy way. So you can look. Each of these bipolar cells can get input from a set of cones, so they will have a receptive field, and now you can think of the horizontal cell as coupling them on a broad.
Is it to have a good model of retina for all?
No, I'm saying this is a first step that you can then take and think of this as individual columns that you'll couple laterally, and you can take this as a starting point for making a spatial model. And I think this is not such a difficult enterprise. Now, how valuable it is, I'm not sure. But you know, I think that if let me give you an answer a la Sompolinsky, is maybe if you have a better way to describe what the retina does, you should do it.
No,
that's what
you had the model, it was validated on flashes of light.
Sure.
That was, one paper, and then you tested that model in a completely different regime in the context of horizontal and not horizontal cell removal, right?
Well,
So,
no, but I kind of agree. I mean,
if the physical model is well tested, then it doesn't need to be choose too much.
no, but
Apply to your domain, and then maybe just adding different inputs to different codes. Maybe that's sufficient.
But I think it depends a little bit. So well, thank you very much. But I kind of agree also with Misha in the sense that I think it depends a little bit what you want to do. So I think if you want to have a model that will reproduce all the possible input-output relations that you have in a given, say, ganglion cell type, and you want to do it for relevant inputs like natural inputs, I think there are a number of ways to do it. This is certainly not the one where maybe the primary way, I don't know. But I think at least thinking about the circuit is certainly one possible direction to develop intuition about how this transformation can happen. I think, for example, as Tim mentioned, this idea that there are subunits that sum non-linearly is not an idea that came out of nowhere. It's an idea that came out of specific examples of ganglion cells that do that from recordings. Now, I don't think my aim was this here. I don't think my aim was to have a model of the retina that can reproduce anything the retina does. I think my aim was to say, if I have a circuit which has some kind of architecture which I can kind of capture in a gross way, and then I perturb one element in this circuit, what kind of changes would it give rise to? And if we have an experiment in which we see many changes, then can I have a way to think about these many changes and make sense of these many changes for myself?
I don't know exactly what your aim was, but I think what you achieved is that you reproduced this particular set of experiments that to me they don't look like very interesting because they are dealing with.
But so I agree with you. I completely agree that reproducing responses to natural stimuli would be more interesting. But I think that by this classification that you have, most of experimental neuroscience is not interesting. You could say the same thing about Hubel and Wiesel, that they looked at responses to bars, and bars have nothing to do with what we do in real life, so the entire field of visual neuroscience is uninteresting. So I agree with you. But it's a criticism, I think, that you could apply very broadly to most experiments in neuroscience.
I'm just wondering if you can do psychophysics experiment giving a brief on pulse or off pulse or brief or long on pulse or off pulse of small amplitude, and if people detect the pulse or not, and if the threshold for on response and off response would be different, and it's as predicted as this kind of non-linearity.
Right. So I think the problem is mouse is very hard to do behavioral experiments, but in fact, I think these kinds of experiments or more complicated versions are actually very much needed in the medical domain for humans, because actually the psychophysics that exists today to evaluate how good vision is extremely, extremely limited and narrow. And developing these kinds of things, just even to kind of have criteria for how good vision is, would be useful.
So what's important is the non-linearity, right, of the cones. Is the non-linearity of cones in mice very different from primates?
No.
Then can you possibly do psychophysics?
Yeah, but then you don't have these PSAM retinas in, for now at least, in primates, but maybe we will.
Dynamics of response to dynamics of psychophysical detection.
Sure.
So what is if the non-linearity is concave or convex? That seems to be important.
Okay, more questions after the
Okay.
I just want to answer to your answer. I think your analogy is very misleading.
Okay.
What was great about Hubel diesel is that they made a discovery, right? They had the cells in the one that people didn't know what would drive them, and they discovered that we use the stimulus and drive them.
Yes.
In the retina, you cannot say that that this full field
So I don't.
stimuli is the
Okay.
discovery of what drives
No, I don't. No. But hold on, Mouse. I think of course I'm not comparing this with any work of Hubel and Wiesel. Of course this is not at all my intention. However, you could say here, you give a full field temporal stimulus and you see that horizontal cells actually matter for responses to that stimulus. So in the same sense, if you want to call it discovery, it's a discovery. Now, it's a discovery of a much lesser amplitude. But why are bars interesting? Why are gratings interesting? That's completely untrue. You don't know what is the optimal stimulus.
They were effective. Before that, people didn't know how to drive the one cell.
But Misha, people study the temporal structure of retinal ganglion cells, and this gives us means to look at the temporal structure, not only the fascia. So Tim focus on the fascia, and this is something complementary. So the full field is means to focus on the temporal structures. That's what we always do.
There's no reason to believe that temporal structure will not be completely different if you apply.
But all these arguments are true for 90% of all the work on neuroscience.
You have to be concrete. 90%. You gave an example.
No,
I think this is wrong.
No, but you always use limited stimuli. That's your point. Your point is you use a small subset of the possible stimuli, and this is always true for any neuroscience experiment.
Okay, thank you.